Welcome to the 2026 Rare Disease Day here in Minnesota. Appreciate the opportunity to be here and to get the ball rolling. So, yeah, whoever hasn't had a chance to grab lunch, please do so, and we'll get started. So, at the onset, I want to thank the hosts for Rare Disease Day. So, the Center for Orphan Drug Research has been instrumental in organizing this and getting this off the ground. And Jim Cloyd, who's right here, and then Brianna, who was up a minute ago, but she's kind of back at the desk now. We're doing a lot of the heavy lifting there. The Stem Cell Institute here at the University of Minnesota has also been a host of Rare Disease Day, and Dr. Walt Lowe, who you'll meet in a few minutes, is in a leadership spot there. So this is me. So I'm Paul Urchard. I'm a pediatrician. I'm in the blood stem cell transplant, cell and gene therapy group here. We've been very interested in cellular therapies as treatments for rare diseases going back 4 decades or so. That precedes me by a bit, not actually that much, but so in terms of Rare Disease Days, this is either the 14th or the 15th annual Rare Disease Day that we've been engaged with. And over the years, we've had a number of different topics that we've discussed. That includes early diagnosis, instruments that are available to help, especially with the genetic diagnoses. Newborn screening is a part of that as well. We talked about therapies that are in development, including gene therapies, clinical trials that are associated with these things, challenges in the trials, and then actually after some of these therapies get licensed, challenges also in delivery and in access. And today we're going to talk additionally about how we develop these therapies, what the trials might look like, and we're going to have several different speakers that are going to address some of these issues. So this is the official designation of Rare Disease Day signed by the governor here. So generally it's the last day of February, but obviously we're doing this here today. I wanted to talk a little bit too about the National Organization of Rare Disorders, or NORD. So I think about 5 years ago, they decided it would be important to create designations of centers of excellence in rare disease. And it's important for people to understand that I think there was 31 or 32 sites that were initially identified as centers of excellence, and Minnesota has 2 of those. So the Mayo Clinic is a recognized center of excellence, and the University of Minnesota is as well. And so that's based on the past history of working with patients that have rare disorders and having a group of dedicated people, including people that are providing therapies and doing research and whatnot in these types of disorders. So, kudos to Minnesota for being prominent in being able to offer these kinds of therapies and being recognized as such. So I think I will then offer this to Dr. Walt Lowe, who's going to be doing the introductions for the speakers and getting the program started. Thanks very much.
Well, welcome to all for attending today's meeting. I think we have an exciting lineup here. I'm Walt Lowe. I'm a professor in the Department of Neurosurgery and the Stem Cell Institute. So on behalf of the Stem Cell Institute, we are pleased to be a part of this Rare Disease Day Symposium that we have supported in the past. Today, I think we have an exciting lineup that deals with 3 areas of orphan drug pipeline, we have regulatory component, we have a researcher, and we have a person here who's a patient advocate. And so I think we'll have, different perspectives of what orphan drug development is all about. Our first speaker today will be Dr. Robert Schuck. Dr. Schuck is the Deputy Director of Translational and Precision Medicine in the Center for Drug Evaluation and Research and the Food and Drug Administration. He'll be coming to us online, so we'll be able to have him present, but also he'll be here to be able to answer any questions that you might have about the FDA. And I'm sure many of you have really concerns about what's happening, and maybe we'll be able to get some answers here. Our second speaker is Dr. Margo Cousin. She's an assistant professor in medical genetics and the Department of Neurology. She's the director of the N of One therapeutics program for the Center for Individualized Medicine at the Mayo Clinic. So she'll be presenting the science component. And then finally, we have Sharon King. Sharon is the Chief Operating Officer of the National MPS Society, and she'll be talking about issues from the patient advocacy perspective. So with that, we will, I should have been showing pictures. So these are the speakers who will be speaking today. So with that, we will turn this over to Dr. Robert Schuck with his online presentation. Can you hear us, Robert?
I can. Can you hear me?
Yes, we can hear you.
Give me one moment to hopefully share my slides. All right, are you seeing the slides on your end, and can you hear me okay?
Yes, yes, we can see them beautifully. So the platform is yours.
All right, excellent. Well, thank you for the invitation. I'm happy to be here virtually. Apologies for not being able to join in person, but as mentioned, I'm Bob Schuck from the Division of Translational and Precision Medicine, which is located in the Office of Clinical Pharmacology and the Center for Drugs at FDA. I'm going to talk about some of our efforts to advance orphan product development through regulatory innovation. I'm going to touch on a few different areas, starting out by covering some of the regulatory challenges in rare disease drug development, moving on and providing an overview of some of the rare disease priorities and initiatives at FDA, and then briefly touch on some of the new pathways and programs that have been announced over the last few months. And I wanted to start out with this, which is, to me, an objective measure of progress. And this shows the CDER rare disease drug approval trends over about the last 2 decades. And you can see over about the last 10 years, there's been a marked increase in the number of rare disease drug approvals every year. And I think this is going to be sustained. And I think this is due to a number of different reasons. I think first and foremost, there's been a lot of patient advocacy, a lot of patient enthusiasm, participation in clinical trials, participation in research that has really helped move the needle. There have been a lot of advances in science, so allowing us to to rely on different kinds of data, such as translational science data, as opposed to strictly relying on adequate and well-controlled clinical trials, and I'll get into that in more detail. And there have been just a lot of advances in enthusiasm in academia, and really the community coming together to figure out what we can do for these patients and how we can better approve drugs. So I do think this will be sustained, and I'm going to touch on some of the efforts that we're making at the FDA throughout this presentation. I wanted to start by going over some of the laws, regulations, and guidances which cover drug approvals. We have these conversations a lot about, this drug should be approved, this drug shouldn't be approved, but it's important to remind ourselves that this is actually based in law. And I want to cover some of the main laws and statutes that guide drug development and approval. So the FDA enforces laws enacted by U.S. Congress and regulations that are established by the agency. And the Food, Drug, and Cosmetic Act, or FD&C Act, is the basic food and drug law in the U.S. And Section 21 of the Code of Federal Regulations, or CFR, interprets the FD&C Act and related statutes. A couple of other important documents. Prescription Drug User Fee Act includes authorization for FDA to continue to collect user fees from applicants who submit certain new drug and biological product applications. This is important because this allows us the funding to review the drug applications expeditiously, and also do some of the important scientific programs that I'll be talking about today that really help us, try to develop better methods and tools for regulatory science. And guidances are extremely important. They communicate agencies' current thinking on a topic. So these are typically written by staff and are meant to help interpret the laws and regulations and clarify how they apply in specific situations. So I'm going to dive a little bit more detail on the FD&C Act here. This law requires that drugs be shown to be effective as well as safe. And again, this is the main law governing approval of drugs. It states that effectiveness must be established by substantial evidence. And this excerpt is a little bit long, but I'm going to read most of it here because it's extremely important. It states that the term substantial evidence means evidence consisting of adequate and well-controlled investigations, including clinical investigations, by experts qualified by scientific training and expertise to evaluate the effectiveness of the drug involved, and goes on to provide some additional information there. So this is very important. This establishes that we need to see adequate and well-controlled clinical investigations to approve a drug. Now, in 1997, the Food and Drug Modernization Act, or FDAMA, amended the FD&C Act to allow demonstrating drug effectiveness with one adequate and well-controlled investigation supported by confirmatory evidence. So this changes the type of evidence that we can use to show that a drug has demonstrated substantial evidence of effectiveness. 21 CFR Part 314 provides additional information on the requirements to submit a drug application. So all the different components that are needed to get the drug approved are in this part of the CFR. And another important act, the biological products are approved under the authority of Section 351 of the Public Health Service Act. And this act states that licenses are granted upon showing that the product meets standards designed to ensure continued safety, purity, and potency of the products, and potency is interpreted to include effectiveness. So this means that biological products are approved under similar framework to how drugs are and are required to demonstrate substantial evidence of effectiveness. Some of the more important guidances covering substantial evidence. 1998 final guidance providing clinical evidence of effectiveness of human drug and biological products. This guidance covers the evidence necessary to support effectiveness, the legal basis and scientific basis for approval, as well as the concept of extrapolation to different subsets. In 2019, recognizing that it had been, a couple of decades since that guidance had been published, and there have been a lot of advances in science, and since that time, we published another guidance called Demonstrating Substantial Evidence of Effectiveness for Human Drug and Biological Products. And this provides more detailed information on things like trial designs, endpoints that can be used, and different things that were, found to be extremely important and we felt needed clarification. And in 2023, there was a guidance titled Demonstrating Substantial Evidence of Effectiveness based on one adequate and well-controlled clinical investigation and confirmatory evidence. And this guidance really provided a lot more detailed information on using that one adequate and well-controlled trial plus confirmatory evidence pathway. So these are, if anybody's interested in FDA's current thinking on drug approvals, these are the three main documents that govern our views on substantial evidence of effectiveness currently. And FDA routinely discusses how companies can develop a drug and meet that substantial evidence bar, as well as all the other requirements for approval of a drug at what we call milestone meetings. So we do interact with companies pretty regularly. These meetings often occur at critical junctions in drug development, so including the pre-investigational stage, so right before a drug is about to go into humans for the first time. End of phase 1 and end of phase 2 meetings, the end of phase 2 being particularly important because that's typically when the design of the phase 3 trial is being discussed and before a marketing application is submitted. And sponsors can also request other types of meetings along the way if they have specific questions about endpoints or something like that. And there are actually specific programs that are meant to do just that I'll talk about for rare diseases. So once FDA receives the application, we conduct our review. And a lot of people, we talk about FDA review of this drug and that drug quite frequently, but I've tried to boil that process down to a single slide here. So it's extremely complex, but this is my best attempt to describe some of the main components of a review. So we're looking at the target condition and available treatments, and this provides a context for weighing the drug's risks and benefits. So we look at a drug very differently if it's, for a minor issue, it's very common, has 10 safe and effective drugs already on the market, versus a drug that's for, serious, debilitating, life-threatening disease, no available therapies, very small patient population that you can't conduct a big clinical trial. And we're of course going to weigh the evidence differently in those situations and take that context into account. We also, a huge effort is put into assessing the benefits and risks based on the clinical data. And evidence that the drug will benefit the patient population should outweigh any risks and uncertainties. So, of course, all drugs have risks associated with them, but we want to see that the benefits are going to outweigh the risks for patients, and we need data to demonstrate that. And then it outlines strategies for managing risks. So drug labeling needs to clearly describe the drug's benefits and risks, as well as how those risks can be detected and managed in order to try to reduce them as much as possible. And I want to get a little bit more into the data that generally supports drug approvals. So this information is taken from the Code of Federal Regulations, which I mentioned is FDA's interpretation of the laws governing drug approval. And it talks about clinical studies. And it describes phase 1 studies as studies that are meant to provide pharmacology and pharmacokinetic information, and states that they typically have about 20 to 80 people in them. Sometimes these are patients, sometimes they're healthy volunteers. As we get into phase 2, these are meant to provide initial efficacy information and proof of concept in patients with the disease, and these typically have no more than several hundred patients. And in phase 3, these studies are meant to provide safety and efficacy information to evaluate benefits and risks, and these typically have several hundred to several thousand patients. So, this is a rare disease group. Most of you are probably looking at this and recognizing very quickly that these numbers simply don't add up for most rare diseases. The drugs that my division covers, typically or frequently will only have, a few dozen patients throughout the country that have them. So clearly we're not going to get a trial with several hundred to several thousand patients to demonstrate efficacy of those drugs. However, it's important to note that the information is still critical. We need to understand the pharmacology of the drug and the pharmacokinetics in order to safely give it to the patients that are going to get it. So we really need to recognize that standard approaches aren't feasible for rare diseases, but we need to find better ways to get this information. And we've made a lot of progress in this space, I think, as shown in the initial slide that showed the increase in drug approvals. There's still, of course, many challenges. I think some of the key ones I've tried to outline on this slide. So in terms of progress, there's increased drug development activity for rare diseases, and it really goes beyond activity. There's increased drug approvals. These drugs are getting approved and being made available to patients. There's more proactive inclusion of patient caregiver voices in drug development. I think we're learning to better understand from patients directly what's important to them, what the risk tolerance is, things like that, in order to help move the needle forward in drug development and clinical trial design. There have been advances in non-clinical models, study designs and technologies, and modeling and simulation approaches. So this gets at the scientific piece. Science has come a long way in the last decade. These new approaches can now be utilized to inform dose selection, efficacy, and safety for rare diseases. And we have precedents for innovative approaches. So we've used external controls to approve drugs. We've used exposure response and non-clinical data as confirmatory evidence. So we're getting confidence in these alternative data sources and still being able to be confident that the drug has demonstrated substantial evidence of efficacy in the absence of some of the large, adequate, well-controlled studies that we would typically see for a more common disease. The challenges that remain, the natural history study is often poorly understood and highly variable. There's genotypic and phenotypic diversity. So, these patients take on very different courses from one to the next, and it's not predictable, and it's hard to capture an endpoint in a study, for example, that is meaningful to patients that's going to be detectable in a study across a number of individuals that have the disease. Drug development tools and biomarkers, such as biomarkers and outcome measures, are often lacking. And there's always going to be a problem of minimal data, particularly when you get into different populations like ultra rare diseases. When you start trying to look at the youngest patients that might get a disease, they're often underrepresented in trials. and different disease subgroups. This is particularly important if a drug has a mechanism that might lead you to believe that a patient would respond differently depending on their genotype, for instance. When you start, you have already a small clinical trial and you start, dissecting that into different subgroups, those numbers get really small really fast, and that remains a challenge. So innovative solutions are needed to facilitate drug development for rare diseases. I like to bucket those into a couple of different categories. Scientific, meaning data sources, trial designs, model-informed drug development approaches, new approach methodologies are all kind of hot topics right now in figuring out how we can improve in these areas to better suit rare disease studies. Endpoints remain probably if not the biggest issue, one of the biggest issues. So validating surrogate endpoints, discussing how we can establish reasonably likely surrogate endpoints within a disease, better clinical outcome assessments to be able to measure things that are important to patients in trials. And from a regulatory perspective, there needs to be flexibility in how substantial evidence of effectiveness is demonstrated, but also consistent interpretation of complex data across different disease states and product types, et cetera. So, we want to make sure, although there's often small differences that might lead to different decisions from one drug program to the next, we want to make sure that the data are being evaluated in a similar way, that, one therapeutic area is not relying on non-clinical data while the other one is saying, absolutely not, that doesn't mean anything, it's non-clinical data. We need to make sure that we have a consistent approach to how we're evaluating these things. And we need to educate stakeholders on regulatory requirements and development and approval challenges so that the community that's highly energetic to help solve these problems can be best informed to do so. I'm going to switch gears a little bit here. I think I've outlined some of the challenges that we face and how we might go about them or what potential solutions might be. I'm going to cover some of the FDA's programs involving rare diseases. And many of these take place under the Accelerating Rare Disease Cures, or ARC program. And this has been around for about 5 years now. This is a CDER program. That is really an umbrella program. It's run out of the Office of New Drugs, but includes all the different disciplines. So pharmacology, toxicology, clinical pharmacology, statistics are all a part of this and work together to work on different programmatic areas to advance drug development. The vision of the program is speeding and increasing development of effective and safe treatment options and addressing the unmet needs of patients with rare diseases. And the mission is to drive scientific and regulatory innovation and engagement to accelerate the availability of treatments for patients with rare diseases. There's a number of different priorities. I'm not going to cover these all on this slide because I'll touch on these in subsequent slides. But engagement and education is one major effort, and then scientific and regulatory innovation. And I'll go through some of the specifics in the next slides. So LEADER 3D stands for Learning and Education to Advance and Empower Rare Disease Drug Developers. This is an educational effort where we've developed some content in the form of educational videos and case examples to help drug developers. The case examples I find particularly helpful. We often get feedback when we write guidances that we need specific examples of how this could be applied for a given drug, and we can't really give multiple page examples in a guidance, but we can do that in this format. So we have, I think there's 6 of these so far that touch on various areas like surrogate endpoint development and establishing evidence of effectiveness. And they really provide some detailed information about how we would interpret various scenarios or how companies that are setting out to develop a drug can do that in the rare disease space. And this is what one of those looks like. It's some screenshots from it. And I'm sharing this for a couple of different reasons. This is for fosdenopterin, which is for a very rare disease. And it discusses the use of a single adequate and well-controlled clinical investigation and confirmatory evidence to demonstrate substantial evidence of effectiveness. And I want to highlight, one, this is really, I think, useful information. I'm not directly involved in these, so I'm not patting myself on the back, but the folks that put these together, I think, did a really nice job. It references specific guidances that can provide additional information. It talks about how you can use the information to establish a plan within a drug development program. And I also wanted to highlight the specific case because it talks about how substantial evidence of effectiveness was demonstrated for this drug. And on the left-hand side, it shows the treated patients. It was 13 treated patients across 3 different studies. And the control was actually an external control of 18 patients. So if you compare this to the slide at the beginning that I shared that outlined the definitions of different clinical trial types or different clinical study types, you can see that this is, very different than that several hundred to several thousand patient study. We're really showing flexibility in how companies can demonstrate substantial evidence and ability to use those kind of alternative data sources like external controls to come to regulatory decisions. So next, I wanted to cover the Rare Disease Endpoint Advancement Program. So this is the pilot program, that joint program between CDER and CBER to support novel endpoint efficacy development for sponsors. It's a pilot that's meant to allow companies to collaborate with FDA on endpoint development, to promote innovation and evolving science by sharing the different learnings in different formats, and to develop FDA staff and capacity and to enable and facilitate development and use of novel endpoints. So again, endpoints are one of the major challenges we see. We have a specific dedicated program to help us work with companies to develop endpoints for rare disease trials. There's a number of intramural and extramural research programs going on. They're all based on regulatory science. So it's things that can help us make better decisions. So a lot of this, again, are going to be in the areas that I touched on already. So things like developing biomarkers, developing efficacy endpoints, how we can use alternative data sources like real-world data and real-world evidence and new approach methodologies in rare disease drug development. And we have various groups at the agency conducting these studies as well as fund external studies as well. I wanted to highlight this specific effort, which is done by my group, where we're really taking a close look at the different biomarkers that can be used in rare disease drug development. So we've developed a series of in-depth reviews on data supporting use of key biomarkers in drug development programs for specific diseases. And we do this to be able to better inform companies on what pieces of data they might want to collect as their drug development program develops. And that way, when we get to the end and we're really trying to make a decision, if there's a key piece of information on a biomarker that would have been helpful, hopefully we have those data in hand and aren't simply, wishing that we had advised collecting the data, years ago before the study was conducted. So it really helps us make informed recommendations on these programs in terms of what information companies should be collecting. There's a lot of translational research efforts. I wanted to highlight the Division of Applied Regulatory Science, which is in the Office of Clinical Pharmacology. So this is a division of wet lab scientists. We have our own lab space. They, work closely with different review divisions. They often attend the meetings with us and things so that they're getting ideas about regulatory challenges, and we can collaborate with them to try to address issues that we're commonly seeing. A lot of their work currently is in new approach methodologies and how those can be applied to evaluate efficacy and safety of products in rare diseases. So just wanted to highlight that this group exists, within a review office at the FDA in order to help us advance the science. To cover a few of the innovations that we've done that directly impact the review of rare disease drugs, I think a few of the most important ones are the translational science team, model-informed drug development, and individualized therapies. So the translational science team was developed because CDER is experiencing a rise in rare disease applications, which often require a deep understanding of basic biology and pathogenesis of disease, and often require substantial analyses of preclinical and early clinical data. And this usually comes in the form of proposals for novel surrogate endpoints or confirmatory evidence, and they require this thorough evaluation that's very difficult to do on our normal review clock. So the objectives of this team are to work with review teams and provide access to subject matter experts on everything from biostats to toxicology to clinical pharmacology and everything in between, that they can use to better assess some of these challenging questions that we need to answer during the course of a rare disease drug review. So the goal here is to aid in improving the quality of the submissions through early feedback, to support knowledge management and consistency, because this team works across all the different therapeutic areas, and also to provide support from senior leadership to review teams on some of these complex questions. Application of model-informed drug development. So MIDD has become extremely common in rare disease or in drug development period over the last decade, I would say, and more recently, it's being applied consistently to rare disease. And it's used for, the most common use, I think, is optimizing dose regimen or informing clinical studies in terms of what dose, should be studied. But more recently, specifically in rare diseases, pediatric dose selection based on extrapolation has been an extremely important use of MIDD. So we often, again, have very few patients under a certain age that might have been exposed to a drug during the drug development program. And we can better model what dose is appropriate for those individuals to make sure that once the drug gets approved, we can approve it for those younger ages and also make sure that it can be used safely and effectively. And then also providing confirmatory evidence of effectiveness. So things like exposure response modeling have been used to demonstrate that the effects we're seeing in a clinical trial are in fact due to the drug and not due to chance. And this has been an important application of clinical pharmacology and model-informed drug development to rare disease drugs. Wanted to touch on individualized therapies a little bit, and this will be, one of the main topics covered in a subsequent presentation. But in 2017 was when the FDA received its first of its kind IND application for the treatment of CLN7 Batten disease that was developed for use in a single patient. And we've had a number of these since. There's several dozen of them at this point. And that led us to develop guidance on administrative procedures. It was published in 2021. And then later in 2021, there were a suite of guidances covering what needs to be done for the chemistry and manufacturing of these products, non-clinical studies and clinical studies. So we've tried to be able to facilitate patients, patient access to these drugs. And that continues today. It was a few years ago that the guidances were produced, but we continue to have these conversations. This is highlighting a RISE workshop from just back in November that covered the current landscape of these individualized therapies, as well as how non-clinical and clinical studies can be conducted, how we can better generate evidence and integrate existing data sources, and what the opportunities will be moving forward. The Rare Disease Innovation Hub is another effort. So this is moving a little bit beyond the CDER programs that I've been discussing now. And this is an agency level effort. Was created as a point of collaboration and connectivity between CBER and CDER with the goal of advancing regulatory science and rare disease therapies through different programs such as the RISE workshop that I just mentioned on the previous slide, and also to enhance and strengthen coordination and alignment between the product centers and to create a centralized point of contact for external partners. So, we've understand that, there's if different things are being interpreted differently by different groups within the FDA, that's not helpful to others. And this program was created to help make sure that we're talking amongst centers and are evaluating things similarly. And the last thing I wanted to cover were a couple of the new approaches to approvals that have been announced over the last 6 months to a year. The first one being the Rare Disease Evidence Principles, or RDEP program. And this is targeted at very small rare disease populations where a drug's intended to treat an inborn genetic alteration, where the clinical course is progressive and there's no adequate therapies. And it proposes a process of using the evidence based on one adequate and well-controlled study with confirmatory evidence to support approval. And the website outline shown below there outlines this framework in more detail. More recently yet, the FDA's plausible mechanism pathway, I've highlighted some information from the New England Journal article that was published by the commissioner and CBER director a few months ago, but I also want to highlight that FDA published guidance on this just last week. I was not able to update my slides, which had previously been cleared for use. But this new pathway covers is meant to apply where you can identify specific molecular and cellular abnormality, where you have a medical product that targets the underlying biological alteration, you have a well-characterized natural history that can be used, and confirmation that the drug was targeted, and therefore can demonstrate improvement in clinical outcomes or course of disease. And I think a subsequent presentation is going to touch on this in more detail. So I just wanted to quickly summarize, I tried to cover a lot of territory in terms of all the different things that the FDA is trying to do in the rare disease space and give a little bit of flavor for the different programs. And I tried to provide links so people can use those to to go back and look in more detail. But I view this as there have been huge advances in clinical and translational science due to work by all kinds of groups, by patients, by academics, by drug companies and regulators to try to move this scientific field forward. This has allowed innovative approaches to drug development and regulatory decision making, and we all have the goal to provide safe and effective therapies to rare disease patients. And I quickly want to acknowledge the staff in the Division of Translational and Precision Medicine, both our rare disease team as well as our genomics and targeted therapy team, make huge contributions to all the programs that I outlined, as well as the Office of New Drugs, Division of Rare Disease and Medical Genetics, and their rare disease team runs the ARC program and is involved in all of these programs. So they really are collaborative efforts that are multidisciplinary, and want to thank all the people involved in these programs. And with that, I'll hand it back to the next presenter. I think we have time for questions at the end, and hopefully I'm within my time. And thank you.
Dr. Shuck, thank you very much for your presentation. So we're going to hold questions for now, and then we'll have a panel discussion where we can answer all of your questions at that time. So our next speaker is Dr. Margot Cousin. She's the assistant professor at the Mayo Clinic, and she'll be talking about from small n to big N, learning, scaling, and transforming rare disease care. Dr. Cousin.
All right. Well, it's good to see everybody. I'm really excited to be here today to talk a little bit about my work, but more to talk about, I think, kind of the landscape we're working in and how we're thinking about really what I have as this title, right? How do we learn and scale and transform rare disease care, especially in this, evolving regulatory landscape. So as you can see, I have the big N made up of all the little N's, right? So how do we learn from our patients so we can move as quickly and safely and effectively as possible? So I'm going to talk a little bit about just, rare genetic diseases and kind of why we study them and how we're approaching the challenges. I'll discuss the small n versus big N concept, give a sort of a brief genetic therapy history. So everything I'm going to talk about today is really geared, targeted around the genetic therapy component. We know that's just one space for therapeutic development, but that's where I work, so that's what I'm going to talk about. And then in the N of 1 gene targeted therapy space and kind of this regulatory evolution and how we can move forward together. So preaching to the choir, I realize with this audience, but just to kind of set the stage, right? Rare diseases are rare by definition, but collectively very common, right? About 1 in 10. Majority are neurologic, often devastating, and the overwhelming majority have no specific, let alone curative therapy. But I think it's important that, the challenges across these rare diseases are often very common, right? And across many different domains. So they all, or most, have barriers that involve limited understanding of the disease with poor models, fragmented data, lack of standards, dispersed patients, difficult to make these trials happen and control them well. Funding is always a challenge. Regulatory wasn't, invented for rare diseases initially, but it's evolving a lot, and we're in a new space now, which I think is great. And then having that infrastructure to be able to move through all of these requirements. So if there's a way that we can address these challenges in a systematic fashion, we can impact a large number of people, right? And that's been my goal. So thinking about our diagnosed rare disease patients, so those that have achieved a genetic diagnosis, let's say, I realize that the diagnostic odyssey still exists for many. But even when we get to that answer of what's causing disease, there's so many other things that need to be done to really improve care collectively. And these are, kind of 5 of the major buckets that I think we need to, kind of check those boxes that we can accomplish. We have to understand that natural history. We have to know how this disease progresses and what that variability is. And now more than ever, we need to understand those mechanisms, at least at the gene and variant level. Otherwise, how are you going to know what type of therapeutic modality can help overcome that? Biomarker discovery and outcomes assessments are going to be critically important to build that evidence to show efficacy, especially in these ultra-small trials. And the only way we can do that meaningfully is to work with our patients and our advocacy groups to determine what are the meaningful outcomes. The worst case is if the patients do actually improve in a number of domains that are critically important to those patients and families, and we didn't measure it, right? Or we didn't measure it well, and that drug doesn't get approved. So that's where the community is really important. And then, of course, the therapeutic development piece, right? We have to charge ahead. So how do we build a reusable infrastructure to systematically address these barriers so that we can get to successful trials, approved therapies, and improved outcomes for our patients? So the N question, small n, and why big N matters, right? So again, N is the number of individuals in your study, right? Whether I'm in the lab working with mice or fish or cells, or working with participants in a clinical trial, N is the number of individuals we're looking at. And really the reason we need a big N, right, is because we want to have that statistical power to measure the positive change associated with our treatment response. That's how we show evidence that this is a meaningful therapy that we want to get approval for. But we understand that in ultra-rare diseases, it's really difficult to achieve that number for many reasons, right? Maybe there's just too few, they're too heterogeneous. And I think with these targeted therapies, is it really ethical to give a placebo to a patient when we know that this is really the only chance they have at a disease-modifying therapy, even if it's experimental? And with these new modalities, right, antisense oligonucleotides, RNAi or siRNAs, gene editing, gene replacement, we can target those single patient mutations. So it's a very challenging space to continue to work in to manage to help those that are in front of us today while maintaining the rigor to be able to give access to all of those in the future. So the challenge, right, convert single patient successes or approaches into a scalable, trustworthy care pathway. So a little bit of history just to kind of set up where we are. I want to talk about kind of how gene and genetic therapies have come around. So 1990 was the first retroviral gene transfer for severe combined immunodeficiency. So a couple of patients in a trial that initiated in 1990. In 1998, we had the first approval for an antisense oligonucleotide therapy. This was for a viral target. Then in 1999, there was the tragic case of Jesse Gelsinger, where there was a severe immune toxicity associated with a gene therapy, and it was fatal. And you know, this really, I think, gave us all a moment of pause to step back and say, how are we, overseeing these studies? What do we really need to be thinking about? How do we do this well and safe? You know, I think prior to this, we had really thought we were part of a gene therapy revolution, and we had to take pause. But things have been moving forward, I think, especially in recent years, quite rapidly. So we had, in 2012, the first gene therapy approval for lipoprotein lipase deficiency. And then in 2017, we had both a CAR T therapy for blood cancers, as well as a genotype-specific gene therapy for retinal dystrophy approved. 2018, we had the first siRNA for hereditary transthyretin-related amyloidosis. And in 2019, the one-time adeno-associated viral vector mediated delivery for spinal muscular atrophy. And I know this was one of the studies that was a really small number of participants, but they had really excellent natural history study data to use as the comparator, and this is often used as an example of how to do these studies very well to get that approval. And then in 2023, the first CRISPR genome editing therapy for sickle cell anemia. Right? So we've spanned from, administering a gene that's been broken in like a recessive condition to replace the functional gene, all the way to really N-of-1 gene editing approaches. So this clinical toolbox, right? And this is just sort of the tip of the iceberg. There are lots more modalities or nuances within this, but right, we have a number of delivery vectors, whether they're viral or non-viral. We can take out cells from a patient and modify those and return those, and sometimes those cells then become the drug in that participant. We think about CAR T therapies as well as some of our gene therapy approaches. RNA targeting mechanisms like antisense oligonucleotides or siRNAs, where we can actually change how that gene is expressed. So we're not actually interacting with the DNA at all or the genome. We're changing the first step away from that at the RNA transcript of that gene. But we can change how it's processed, how it's expressed. And then genome editing. Can we go in and make those very precise changes to maybe correct a mutation or impact a meaningful change? So again, kind of three main buckets maybe we might think of where we have diagnosis specific. So maybe it's by gene, or that patient batch where we're actually taking out cells, modifying them, or that's a living drug in that patient, or maybe an N of 1 design where we're correcting a very specific alteration. But what's been making this possible is the fact that we're actually getting genetic diagnoses at a much better rate than we ever were in the past. So our sequencing and variant interpretation is far improved. Better delivery systems, whether those are viral vectors, lipid nanoparticles, or improved chemistry of our other modalities. And manufacturing, and this is not a small space, right? So I didn't realize early on that actually manufacturing small batches is a significant challenge. So we're used to making even like antisense oligonucleotides for the whole population that needs them, but what if it's just one patient? How do you cost effectively manufacture a small batch at the same kind of rigor that you would a large batch? So they had to revamp really how those manufacturing processes happen to do that well and do it efficient and cost effective. And I think a lot of industry groups are investing in those processes knowing that this wave is coming. And again, the regulatory pathways are continuing to evolve to allow us to move forward in meaningful ways for these ultra-rare disorders. Persistent constraints, right? There are many, I have a few here, right? So delivery to the tissue that really needs to have the drug is still a challenge, right? We can deliver to some well, everything goes to the liver, liver's great, but muscle's a little harder, cardiac's a little harder. But many groups are working on that. I don't think that will be an insurmountable challenge forever. Safety, always a big question, right? Immune reactions, off-target effects, integration risks, always need to be top of mind. So as we move forward to maybe future state where some of these processes might become the drug that needs to be built in, right, to really assess for safety always. And then, without a doubt, cost, access, and complex care pathways will forever be a constraint in this space. Finding ways to come together to be mindful of those will always be important. Okay, so just to kind of summarize, our gene-based therapy approaches, right? We have DNA-directed therapies. We think about gene transfer or gene editing approaches where we're actually engaging with your genome or providing DNA. And then the RNA-directed strategies. So that would be antisense oligonucleotides or small interfering RNAs, where again, we're just changing how that gene is expressed or how that gene product is coming together. And I always have to go back to Tim Yu's work on Mila-Sin. This was published in 2019, and I saw him give this talk at the American Society for Human Genetics and realized this is what we need to be doing. So Tim Yu was able to develop a custom antisense oligonucleotide drug for a little girl named Mila who had Batten disease. Using the same chemistry that is done for nusinersen, which is an ASO for spinal muscular atrophy. So use the exact same chemistry, just change the order of those nucleotides to target a region that was going to be beneficial for Mila. But they were able to work with the FDA and get this to a first dose of her trial within less than a year. And I realized that's a hammer I can swing maybe coming out of genetics my whole career. I can think about medications in that way. I can think about how this might move forward. And this did provide benefit for her, reduced seizure burden, increased quality of life, and changed, I think, the way we think about individualized medicine, especially for rare diseases. So just one slide on really why ASOs. This is a space that I work in a lot just because of the opportunity that exists today. So again, these are antisense oligonucleotides, so short strands of modified RNA that interact with the RNA product of that gene. And as Dr. Schuck mentioned, there's now guidances for how to develop these and translate these for individual patients with these severely debilitating and life-threatening conditions. And I think the concept that's really important here, and I think for some of these N-of-1 approaches moving forward, is that we know what the risk of not treating these patients are, right? They're dire, they're terrible. These patients have severe disease. And we also are really getting a good handle on these chemistries, right? We understand, how these ASOs and other drugs work, the safety profiles, and not treating is riskier than treating with a first-in-human experimental ASO in these cases. So how do we make sure that we're still being rigorous and safe, but move at a pace that we can actually address the needs of these patients? ASOs have other attributes that make them, I think, a good opportunity for N-of-1 in that they're delivered as naked molecules, so I don't need viral vectors or lipid nanoparticles or complex packaging. They're extremely stable, so they can sit on the shelf for decades. We can make that single batch, and that patient can draw on that over the lifetime that they need that drug. So relatively cost-effective to synthesize. The effects are temporary, right? Pro versus con. You have to repeat dose. But that also means we can start at a really low dose to make sure it's safe. We can slowly dose up, we can adjust as needed, and we can discontinue as needed, or when something better comes along. So it gives us quite a bit of control in how we would manage that patient. And then because of that, there's been some community resources that have become available. So the NLORM Foundation was started by Stan Crook, who's the former CEO of Ionis, to develop ASOs for individual patients. And they're doing this out of charitable donation and providing these drugs for free for the life of the patients they take on, as long as we think they're achieving benefit. Amazing opportunity. We're working with them, as are many other groups. And the N=1 Collaborative was started. So this is an academic consortium. I encourage you all to look into them if you haven't, that are trying to define best practices and really and provide resources, but build the community so we can understand how to do this the best way. So we can learn from each other and also build the data sharing platforms so that we can get from these N=1s to learn for that big N, right? So can we get to process as the drug as opposed to every single molecule having to walk through this process independently? And then the story probably all of you know about, right? Baby KJ, last year, the first in vivo gene editing therapy. And the timeline again here is quite remarkable. And the Herculean effort it took to get this from genetic diagnosis to therapeutic dose in 8 months is truly incredible. Clearly, this process is not going to be sustainable. So how do we learn from this, and how do we build and say, you know, what worked here, and where are the places that we need to build efficiencies? You can see Kieran and Rebecca here holding baby KJ, and I hear he's doing quite well. So from this, though, Rebecca and Kieran and others have said this is not how we're going to do this. We're not going to keep repeating that process as we did it the first time. So they've had conversations with the FDA and try to figure out, you know, what makes sense. How do we have, you know, get approvals, or think about, you know, we can reuse the data from our lipid nanoparticles and our editor that's already been now administered clinically, and just think about what we need to do to show safety of the new guide RNA and the new target, for instance. Can we get a primary IND with a master protocol and then have sub-protocols that reference that so we can move quicker and say we're going to do the process the same way, we're just changing out, you know, the patient and the target. Maybe it's in the same class of disease. So really trying to be thoughtful about how we can make best use of resources and move quickly to scale. And then, as Dr. Shuck mentioned, the plausible mechanism framework draft was just released last month, which is really exciting and quite a buzz in the community for sure. And I think it finally gives us a path and a roadmap that we can start to plan for, right? You know, we have to know the target, right? What is the genetic or cellular or molecular alteration we're going to target? We have to know we're targeting the pathogenic pathway, right? So the known mechanism of disease. We need to have that natural history data. We have to be able to confirm that we are drugging and editing in the way that we think we are, and we have to demonstrate clinical benefit, right? So not all of us are going to be able to tackle all of these things, but as a community, we can really start to hammer out those parts that we're going to need when you're ready to engage in this pathway. And I think that's the message here, is the community coming together to solve these and trying to do it in a systematic way, right? How do we do natural history studies more systematically? How do we reach our clinicians where they're at with their patients so the patients don't have to enroll far away to share their data for the common good here? Tim Yu and Mila's mom, Julia Vitarello, and other colleagues who participated in baby KJ stories and others had a response to the FDA's proposed pathway, and I wanted to share some of their thoughts here. Right? How do we go from bespoke to scalable? Obviously, there is an urgent need for a sustainable path. Glad to have a path at all, but sustainable is going to be a need to really scale. And again, these concepts of a single umbrella IND, which would then facilitate maybe this process being what's approved as opposed to each individual molecule or drug. Can we group mutation or technology classes across diseases in basket trials to try to show that the platform is safe and we understand what those potential side effects and risks are to move forward? And again, standardize the full process. So this starts from variant identification, design, manufacturing, in vitro performance, preclinical safety, all the way through the clinical administration and monitoring, right? If we can say that this whole process will be done the same way every single time, these patients will be treated the same every single time, then those data can be used collectively to inform decision-making down the road and hopefully the approvals. And I think there needs to be flexibility in how we're going to demonstrate efficacy, and that's going to be very dependent on those patients and the diseases, right? Some of them are more variable than others. Some patients are going to be on their own trajectory, so maybe it's a pre and post-treatment for an individual patient comparison, less about cases versus controls. So I think coming up with strategic ways to measure efficacy and show it rigorously is going to be important. We need to maintain the rigor, but we need to find ways to show that works with our patients and then how their diseases manifest. So then after a series of interventions, right, can we estimate the probability of response and that adverse event profile so that we can submit that marketing application? And then I really like this piece, right? So after the approval, we need to make this a learning system. So this really speaks to, again, the process and also pooling that data together so that we can all learn from it. The negative data are sometimes even more important than the positive, right? What didn't work? Why didn't it work? Can we learn from that? Especially to make use of these really individualized data. So can we establish then within this process that predefined patient criteria to say this type of genetic alteration in this type of disease with a patient that has this therapeutic window, we can put into our program without having to go through that huge review every time, going back to the FDA every time to say that no, this is what works. This is what we've seen work because we've done it now in a large trial of many different patients. And then again, build that centralized evidence base so we can learn from those data. And then I think planning reimbursement early, I mean, again, preaching to the choir here, I think today, but making sure we're thinking about our payers and working with them and what they need to see in order to participate is going to be key, again, to scale. Okay, so that was a lot. So what do we do now as a community? And again, I think it comes down to all of those, individual checkboxes and coming together to do it systematically. There are groups that just put forth, again, this Herculean effort to solve one problem, and then the next group has to do the same and the next. So, can we streamline these approaches collectively to, combine patient populations, to figure out ways to have better functional assays that we can use, because that mechanism is going to be important. Not only do we have to know it's pathogenic, we have to know why it's pathogenic and what it's doing, so we know how to fix it. Putting that natural history study data together working through our partners in industry to manufacture and administer these drugs, and again, the learning piece. So coming up with the protocols across institutions to share data that we know our patients often desperately want to share, and our regulatory groups often prevent us from doing seamlessly. We need to find a way to do that so that we can move the needle forward together. All right, I want to share a little bit about what we're doing at Mayo Clinic in this regard. I don't have a lot of data, but just kind of more the processes and how we're thinking about these issues. So I'm the director of our N-of-1 Therapeutics program in our Center for Individualized Medicine, and there's kind of a lot of words on here, but essentially we're trying to streamline at each of these steps a process flow so that we can get our patients where they need to go. We don't have the resources to take on every patient program and develop a drug for every single one, but if we can at least facilitate understanding what it is that they need and connect them with resources that are maybe outside of our institution, if that's the case, then we can really move things along quickly. So going from genetic diagnosis to, kind of an automated review of currently FDA-approved medications for that condition, currently available clinical trials, and then doing that novel therapeutic modality matching. So is this genetic alteration patient a good candidate for an ASO? Or maybe it really needs to be, edited precisely because of the nuances of that gene. Maybe there's drug repurposing approaches, which I haven't really talked about much today. And from there, can we then discuss those cases and figure out what we would communicate with that provider or that patient, and what opportunities we think are appropriate to suggest, right? So we have an interventional genomics review board to review those cases and suggestions, and also help us make use of our very limited resources on those patients that we might take forward with efforts internally. And then where do we actually develop these therapies, right? So the Cousin Lab is doing a small amount of work in our small space, but largely these are very collaborative approaches. So if I know someone's studying this disease, I'm going to call them up and say, hey, we have a patient that I think that could really benefit from your work. We're working with the N-Lorem Foundation and other external collaborators. So again, community, it doesn't need to be a siloed effort to get patients to opportunities. And then the delivery. So we're working with our Office of Clinical Trials to think about how to streamline these approaches, to use resources effectively, to give opportunities to our patients in a way that we can, again, save our resources as much as possible. And then on the left there, establish this clinical trial readiness. So we're working with our patient advocacy organizations and patient populations and collaborating with groups that have built infrastructures already, if they exist, to do the natural history studies, to contribute our patient data, to collect samples, to do biomarker development. So trying to systematically build that infrastructure and that collaborative network to be able to plug our patients into those opportunities. So with that, and in partnership with the N-Lorem Foundation, we have a handful of patients that are either receiving experimental ASOs or are in the process of drug development. I've been partnering with Dr. Brendan Lanfer, who's been my clinical partner in developing this program. So we have an IRB that allows us to share with external groups data and samples for the purposes of individualized drug development. And we have agreement with N-Lorem to work with them. And one of our patient trials, it's been a little over a year now, a patient with autosomal dominant leukodystrophy. So this is caused by a lamin B1 gene duplication that leads to overexpression. So we're knocking down that expression with an antisense oligonucleotide. No known biomarkers for this disease. There hasn't been a natural history study. So we're sort of, building the plane while we're flying it. Neurofilament light chain is a biomarker of really neuron degradation and neuron injury. So not been proven in ADLD, but his prior to dose 1 levels were extremely high, and after through dose 4 we see them drop precipitously. So signs were at least going in the right direction, but it's way too soon to tell whether we actually see clinical benefit. But with this, we're trying to check all the other boxes, right? So we're partnered with the ADLD Center, which is the patient advocacy organization. We've been working with them for several years to help us define needs, goals, direction. We've put out a clinical practice guidelines paper and are collaborating with the other leaders in the field in this disease. Partnering with the N-Lorem Foundation to bring an ASO forward and are hoping to expand that trial this year. We're working with Children's Hospital of Philadelphia and the Global Leukodystrophy Initiative and leveraging their infrastructure to do our natural history study. So we're leading the study, but leveraging this beautiful database system that they've built as a part of that group. And trying to expand to a prospective study, biomarker development, and participate in additional therapeutic development opportunities. So this work has been in collaboration with Drs. Lanfer, Tobin, and Demeja at Mayo, who have been instrumental in moving these programs forward. Okay, so build the big N. We need the evidence that justifies the risk and the cost. So to scale access, we need this evidence that's going to be credible to all the key stakeholders. And again, it doesn't need to be a single huge trial. It can be high quality evidence across many of these small N's. So again, the harmonization and shared approaches across all of these domains are going to be critical, and I think that's the charge I would put forth to this group, is to continue to shake hands with your neighbors and see who can help you get across that finish line the most rigorously and quickly that we can do. And part of this is going to be new business and practice models, and I'm certainly no expert in this space, but we've been having a lot of conversations with different groups and thinking about sort of this N-Lorem interesting space where it's really a nonprofit biotech partnering with clinical institutions to get novel experimental therapies in our most needful patients. If we can get some of those to approvals, how do we create the system where the royalties from those can feed back for the next ultra-rare disease? Not feedback to pharma for the next common thing where they want to develop another blockbuster drug, right? How do create the ecosystem so that we can crank the flywheel and it builds upon itself so we can do more with the small amount of maybe reimbursement that we might get for a single patient trial or a single patient drug? Centers of excellence approach, I think this is also a really interesting concept in that there have been a couple of different centers for genomic surgery that have been coming up right? Thinking about, again, that this is really more of like a high-risk surgical approach than it is really a typical drug in the sense that we would often think of. So, can we get centers of excellence that might have some sort of accreditation in a way that maybe a really high-risk surgery might, where we can deliver these genome editing type therapies? Then maybe it follows a different sort of approvals, regulatory processes that might be, again, more like a high-risk surgery, also maybe kind of like a lab-developed test as far as how we manage those processes. So I think it's a novel way to think about these, as it's not so much like a small molecule that we're giving. And I think these philanthropic and public-private partnerships are going to be key to a sustainable future. So takeaways from Path from Small N to Big N, again, platformize the parts that repeat. Use standardized pathways to decide when N of 1 is scientifically and ethically justified, but do our best to make sure that that data come together so we can learn together. Invest in the infrastructure so that, again, this can be repeatable and systematic. Continue to partner. The rare disease community I found to be absolutely amazing, and the people who are here really want to do it for the right reasons and are largely collaborative. So let's continue to build that ecosystem. Design for access from day one. Let's do our homework to get biomarkers, outcomes, and safety data that payers can trust and create sustainable models to continue to move forward. So again, can we come together and solve these challenges across all of these domains so that we can get to improve patient outcomes across the board for our rare disease patients? Huge ecosystem that we work in, so I want to kind of throw everybody up here, but really pleased to be here today, and thank you for your attention.
Thank you so much for your presentation, Dr. Cousins. So our next speaker is Sharon King. She's the CEO of the National MPS Society, and she'll be talking about when purpose aligns, patient science, and shared resolve.
It's not coming up. There, wrong one. There we go. Thanks. Thank you. Start with a technical issue, but thank you for that. So I am so honored to be here today and join my fellow panelists. As both a longtime advocate for rare diseases and the mother of a child who lived with a rare disease, I deeply appreciate the dedication that each of these panelists bring to this community. And this is a community that I truly call family. Your commitment and your tireless efforts mean so much to my family and me, and I am sincerely grateful for all that you do every day to support and uplift those affected by rare diseases. And I want to thank you for making a difference for us. During my time as senior advocacy lead at Aldevron, we partnered with the University of Minnesota's Center for Orphan Drug Research to bring the 2023 theatrical production, Rare Stories of Disease, to the iconic Fargo Theater. And Dr. Cloyd, it is such a pleasure to work with you again and to be invited to be a part of this panel. And I don't know that he's here, but I also want to recognize Dr. Chet Whitley. My connection with Dr. Whitley goes back a few more years. I attended my first World Symposium, which is an annual international research conference dedicated to advancing the understanding, diagnosis, and treatment of lysosomal storage diseases in Orlando, Florida in December of 2006. Dr. Whitley had just founded the conference, I think maybe a couple of years earlier, and it was already noted for me as a must-attend a conference of a mother whose child had just been diagnosed with a lysosomal storage disease. But finally, I want to honor the millions of individuals living with rare diseases here today, as well as the loved ones and the caregivers who stand beside them. They are the very reason that we're gathered here today. It's the beautiful souls like these children and families who are living with MPS that drive my work every day. And it's essential to remember that rare diseases do not affect just the person diagnosed. Their impact extends to the moms, the dads, the siblings, the grandparents, all of those who also endure the pain and the uncertainty that often comes with a diagnosis that is lacking available treatment. These patients and families demonstrate extraordinary courage and resilience, and they share a profound wisdom that is shaped by their journeys. And it's the kind of wisdom that only the hardest experience can impart. If you are a rare disease patient or caregiver, I hope you know that we see you here today, and importantly, we hear you. If you see that we may be forgetting to listen on occasion, please speak louder. Your stories must be heard. They are the stories that become our purpose. So I want to go back to my first World Symposium and why I was there. Just 5 months earlier, my husband Jim and I had received the very worst news we could have ever imagined. Our youngest daughter, Taylor, was not quite 8 years old and diagnosed with CLN1 disease, which is a part of a group of conditions commonly known as Batten disease. They're progressive, degenerative, lysosomal storage disorders. That moment on July 24th, 2006, when the geneticist shared the news with us, is just burned in my memory. I can still see the way the sunlight danced in patterns across the floor of his office as he told us that there was nothing that could change the outcomes for her condition. We should enjoy the time we had left with her and make happy memories. And I have to tell you, as I watched that sun dancing across that floor, I remember thinking, happy? Will I ever have another happy moment in my life? The devastation in that moment was so real and so complete. I'm not sure how we even walked to our car. I know that we couldn't drive home. We couldn't find the place to put the key in the ignition. It wouldn't have been safe to drive. So we sat in our car on a North Carolina July day. And for any of you who have been to North Carolina in July, you know the heat and humidity is stifling. And we just sat in that car. We were stunned. We were shocked. And there was complete silence for about an hour until, there were no words until there were. I'll have to tell you, and Jim would probably tell you, that after about an hour, I pounded the dashboard of his car, and I swore that I could not watch that happen to my child, the description we had just received. We could not watch that happen to her without a fight, and I was going to go to war, and I keep my promises. I lived and breathed finding a treatment for CLN1 disease. I'm still living and breathing it. Taylor's passing in 2018 marked a devastating loss, but it didn't diminish my dedication to supporting children and families facing a Batten disease diagnosis, and my commitment has always extended beyond Batten disease. From the very beginning, I recognized that advancing progress against rare diseases demands collective action. We're not going to get very far if we work in isolation. True progress comes from the strength we find in coming together as a community, uniting all of those impacted by rare diseases. In that shared purpose and connection, we have the power to overcome the greatest obstacles in developing treatments. Yet it's still hard. We must shift our perspective on rare. For a long time, I want to tell you, rare was a 4-letter word in my house. Somebody said rare, and I went, no. Rare leads you to believe that it happens infrequently. Why should someone care if it doesn't happen very often? Rare happens really often. Thank you for bringing that up. I choose to define rare as unique and worthy of our unwavering focus and investment. I know that everyone in this room appreciates the significance and the insights gained from studying rare diseases, but does the broader public truly grasp what is at stake when we view rare as not occurring very often, and as a result, deprioritize the sustained commitment and the resources needed to find answers? This mindset risks leaving too many families without hope, and it's up to us to challenge it. If you put 2 patients in a room, the conditions they face may be very different, but their experiences and the stories they tell are always quite similar. And without fail, they share a common desire, a treatment that will bring a better quality to their life. In my almost 20 years as a rare disease advocate, I've seen good, really remarkable science, and I've seen it go nowhere. And why? Because we can't get science over the roadblocks that exist beyond discovery. We've talked about the Valley of Death for years. Taylor's Tale is the public charity that my family and friends started after Taylor's diagnosis. We invested in a video describing these roadblocks to getting drugs to rare disease patients. Roadblocks driven by small, dispersed patient populations, regulatory hurdles, and extreme economic pressures. I think that video was created sometime in the 2017 timeframe, and we're still talking about those same roadblocks. Nearly 20 years have passed since that pivotal moment in the parking lot of a geneticist's office. And in that time, significant strides have been made toward developing treatments for the 13, and some will say 14, forms of Batten disease. In 2017, Brineura, an enzyme replacement therapy for CLN2, received approval and remains the only approved treatment for any form of Batten disease so far. It's progress. It was a really big deal for the Batten community, but it's certainly not enough. And CLN2 families will tell you that they want more too. As a Batten disease community, we often find ourselves nearing breakthroughs, only to encounter setbacks. These obstacles are not always rooted in scientific challenges. It's the other roadblocks. Taylor's Tale began supporting the development of an AAV9 gene therapy in 2013, and in just over 3 years, the therapy was licensed to industry. It earned both the orphan drug and rare pediatric disease designations, and in May 2019, the FDA announced clearance of the IND application. Although Taylor had passed away just months earlier in September of 2018, our drive remained strong. We felt as though there was an angel guiding us from above, and yet, after that milestone, progress stalled. The silence was like an always ticking clock, and it was deafening for the CLN1 community. Starting in January 2020, I began searching for answers, and by August, the program was sublicensed to another sponsor. This new sponsor, committed to advancing the therapy, and clinical trial enrollment was anticipated for spring of 2022. However, in February of that year, I learned that the program had been paused as investment had slowed. You can see what has happened over a number just recent years for the Batten disease community, starting in 2021, looking at 2023, how investment and focus had slowed. During this loss of momentum, Batten disease, the CLN1 community was not the only Batten community facing significant setbacks. For the 12 active programs in 2021, there continued to be no approved therapies for any form of Batten past CLN2 disease. As you can see, efforts to revive progress are now emerging, but the path forward continues to remain unpredictable and challenging. This uncertainty is leaving approximately 14,000 children without viable options. And while this number may seem small on a global scale for any parent, one child, their child, is enough to declare enough is enough. Children and families affected by Batten disease deserve access to effective treatments. So because of this, I really can imagine how the Muldoon family felt when their newborn son, KJ, was diagnosed with CPS1 deficiency, a rare urea cycle disorder that prevents the liver from breaking down ammonia, causing toxic buildup that can result in permanent brain damage or death. KJ needed a liver transplant, but his parents were offered a different solution, an innovative, transformative approach, and KJ would become a pioneer. Just 6 months later, this little boy received a gene-edited treatment tailored especially for him, and today he's thriving, and while he's not cured, his condition is now much milder. The world closely follows KJ's journey, celebrating his milestones like his first steps with his family, made even more meaningful given the obstacles he and those who love him have had to overcome. I was able to meet KJ this past week at Rare Disease Day at the NIH. In the final session of the day, Drs. Kiran Musunuru and Rebecca Ahrens-Nicholas and KJ's mom, Nicole Muldoon, were on the stage sharing their experience with the audience. I had a flight to catch, so while I really hated missing that last session, I had to leave. So I dashed out into the auditorium to grab my bags out of the auditorium, and sitting on the steps, right as I exited, was KJ and his caregiver. I was able to talk to him a little bit, and he is absolutely your average, cutest toddler you can imagine. And yes, that smile really can melt. I'm sure it would melt even the hardest heart. As I raced away to grab my bags and head to the airport, I kept thinking, oh my gosh, science is wonderful. And I followed up with, and we need the will to make science work for us more often. KJ represents hope and possibility for the future. During my time at Aldevron, one of the partners in the quest to bring a treatment to KJ, I witnessed a powerful public-private partnership transform tragedy into triumph for the Muldoon family, demonstrating what's possible when patient needs, science, and determination come together. After the celebratory announcement at ASGCT at the annual meeting in New Orleans this past May, I found myself reflecting, what's next? Where will we go from here? I knew families like mine would see coverage of this life-changing therapy and wonder, well, why not me? Why not us? Why not my child? The question is not if, but when. When will the next breakthrough happen? Children like KJ, and yes, children like Taylor, who do not have time to wait, need therapies now. I want to share a quote from Dr. Ahrens-Nicholas, the physician who delivered KJ's personalized therapy, and it captures the heart of the challenge. We need to be able to scale this in our country to do that. You have to have an approval so that the drug product can be reimbursed and we can actually subsidize this to be able to help patients around the country. The technology is there. We just need the infrastructure to evaluate it and deliver it. That same need applies to the gene therapy that Taylor's Tale has been pursuing for over a decade. I've said many times that it feels as if the science is the easy part. And I know for all of you researchers in the audience, don't throw stones. I know how hard you work every day. But it's all those steps and those actions that come after the science. I am so hopeful that Drs. Musunuru and Dr. Ahrens-Nicklas will receive the approval for a formal clinical trial that allows for these multiple individualized treatments to be tested in a single umbrella trial. After treating 5 patients, the team would plan to move to a phase 3 trial, which could enable conditional support and an approval of base editing as a platform technology. I'm really interested in this path to a platform, and I've long been an advocate for platform technologies. I hope that we'll keep our foot on the gas pedal for this one. I was in Washington, D.C. in 2015 for a meeting at the NIH to discuss North Carolina's newly legislated Rare Disease Advisory Council. It was the nation's first. And someone shared the draft of a paper around platform approaches for AAV gene therapies. And I would later go on, have the privilege to serve on committees within the Bespoke Gene Therapy Consortium. I was at the NIH and received that draft in 2015. That was 11 years ago. I was once told that moving good science to patient takes 15 years. Maybe that is truly the case, but our children don't have that long. We need something now. In a world of uncertainty and discussions around risk-benefit, one thing is clear for many rare disease patients and families. The risk, the result of doing nothing is certain death. For individuals facing progressive illnesses, every day counts. Nicole Muldoon was very clear in her comments at the NIH as she and KJ's dad were considering the best path forward for their infant son. They were also considering the understanding that would be gained for future children, not only those who would be diagnosed with CPS1 deficiency, but other rare diseases. I promise you, families get risk benefit, and they know very quickly their place within a larger community. They are family. And while we're at it, can we please find a way to ensure that children are diagnosed early enough to benefit from these scientific advances? Many infants with CPS1 deficiency, that most severe urea cycle disorder, die within the first week of life. And Dr. Ahrens-Nicklas made the point very clear last week that the action that truly precipitated saving KJ's life, making it possible for him to receive his pioneering treatment, was the biochemical diagnosis of CPS1 deficiency, enabling the clinical team to get him on dialysis within 40 hours of birth. So I want to share a little about the MPS perspective. 3 weeks after the baby KJ announcement, I took on the role of Chief Operating Officer and Advocacy Lead at the National MPS Society. The MPS community has been a guiding light in my own journey, and it feels natural to champion the cause for kids and families affected by mucopolysaccharidosis and mucolipidosis. Again, serious, progressive lysosomal storage disorders. These patients and families are searching for hope and a better path forward, a treatment that can truly improve their lives or the lives of their loved ones. While patients and families need support, What they want is a treatment that makes a meaningful difference. At the National MPS Society, our mission revolves around the principle of drug to child. Everything we do is driven by the urgent goal of getting therapies to those who need them most. We are an organization that tackles challenges head-on, and we will not shy away from the difficult task in the fight for better outcomes. We do hard things. Thanks to advances in research, several types of MPS now have access to first-generation therapies. Last summer's family conference offered a powerful firsthand look for me at the life-changing impact these treatments provide. During the event, our CEO and president, Terry Klein, invited me to present at our Spirit Conference, which is a special one-day conference that rests within the larger annual family conference. And it's designed for our adult MPS community. The Spirit Conference is dedicated to supporting adults living with MPS and ML. And as I approached the podium and I looked out over the audience, it washed all over me that these were people who were in that room because they had an available treatment. MPS is typically a pediatric disorder. Many attendees in this room were thriving, and they're well into their 30s. There were a few 40s in there. Clear evidence of how treatments can transform disease outcomes. Witnessing science actively enhance lives with improved health and brighter, more stable futures. You cannot imagine, for somebody, the new kid on the block in this organization, you cannot imagine how inspiring and reaffirming it was as I embraced my new role to see what can happen. At that same family conference, I connected with families whose children are participating in clinical trials for promising second-generation therapies designed to cross the blood-brain barrier and slow the cognitive impacts of some forms of the disease. Tragically, several of these studies are facing significant challenges, leaving families grappling with heartbreaking setbacks. Each delay may mean a child loses the ability to walk, to talk, or even recognize mom and dad. The urgency for our community is palpable, and for many of our children, every moment counts as we race against the relentless advance of disease progression, because we know that time lost is time that may never be regained. So like Batten disease families, the diagnostic journey can be very long. Families watch their child regress, and while their concerns are often dismissed as, well, every child develops differently, or let's just wait and see what happens. And all the while, we're losing precious treatment windows. They're closing. Brain damage is occurring. Skills are being lost that we may never regain. The National MPS Society has championed 2 successful newborn screening nominations for MPS I and MPS II, and approved by HRSA. Babies born are being screened nationwide for 2 of our 12 MPS conditions. But I gotta tell you, the 10 that are missing is 10 too many. From the MPS perspective, 3 things must happen. The first is we need early detection. Let's get that right. We need available blood-brain barrier crossing treatments, and we need immediate access. We're failing families and children on 2 of those 3 requirements. We understand that rare diseases require fundamentally different approaches because traditional regulatory and business models were built for common conditions with large patient populations and predictable endpoints. Batten disease and MPS don't fit that mold. And while we wait for the scale-up of new technologies like the one that has transformed KJ's life. We need something now for today's children. We can't run 5,000 patient trials. We don't have 5,000 patients. We can't wait for 10-year survival data when children are dying in less. We can't use traditional endpoints when the disease affects multiple organ systems differently in every patient. Congress realized this when they gave the FDA the power to move treatment for rare diseases forward through the accelerated approval in the 21st Century Cures Act. Both delayed treatments for forms of MPS have been in development for over 10 years. And our families are thinking, what's accelerated about that? Yet, the science is undeniable. We've seen gene therapies stabilize children who would have died. We've seen enzyme replacement therapies give kids years they wouldn't have had. And we've seen improved outcomes, children who can walk, can talk, and live, while their siblings with the same mutation could not. We know these children. We need a sustainable regulatory ecosystem for rare diseases now that is accepting of different evidence standards. Small trials, natural history comparisons, and patient registries as real-world evidence. We need acknowledgement that placebo-controlled studies for our populations are unethical. In rapidly progressive pediatric brain diseases like Batten disease and MPS, we need the FDA to accept disease-specific biomarkers as evidence that therapy is working. Biomarkers directly measure the toxic buildup destroying children's brains. When levels decrease, we see proof that treatment is reaching the brain and stopping damage. Rejecting biomarkers in devastating pediatric diseases doesn't protect patients. It guarantees irreversible loss. And without biomarker acceptance, proven science becomes more difficult to advance. So I want to be clear. For us, this is not about lowering standards. It's about appropriate standards for a devastating disease, while doing nothing means certain death for our children. We cannot lose another generation of children waiting for a more perfect solution, nor can we accept the investment crisis that's being created. When science works, but uncertainty is high, biotech pulls back. Maybe it leaves us. It goes outside of the U.S. The fact was clear in the earlier slides showing the effect of delays on the Batten community. We need policy that reduces regulatory uncertainty and ensures innovative investment continues. So finally, I want to go back to baby KJ. From the patient advocacy perspective, innovation requires partnership and urgency that matches the science. It worked for KJ Muldoon, and it must work for other children needing a chance at a better future. Please keep the gas pedal to the floor. We must sustain investment through and over the hurdles that are undoubtedly going to come into view. Our kids need treatments now, and please don't throw out the really good as we pursue the next iteration. The result's going to be another lost generation of children. Science iterates, and we'll continue to find better and better. That's what innovation's all about, and that's what we do so well in this country. We need frameworks that encourage investment in innovation, especially when early data is promising. It's been 19 years since Taylor's Tail funded an enzyme replacement therapy, 13 years since we funded discovery of a gene therapy. I've watched several generations of children lose their lives waiting for the opportunity. I can call them by name, many of them, and it makes my heart hurt. So I'm not going to read through the list with you. There are 23 rare disease programs that received CRL since early 2025. The MPS community, especially our MPS 2 and 3 communities, have been shattered by these delays. And here are some of the facts and effects that have them reeling. 1.74 billion invested in programs with an accelerated approval stalled at the finish line. Programs that have been in development for more than 10 years. And for a family, accelerated means we're going faster. 10 plus years. We need to move faster. 54 children on hold and more in the waiting and the wings. 2,500 families impacted by these decisions, and children continuing to progress past the point where the drug is going to make sense for them. So we need to better, and we also need to better define access. I want to note that true access is more than an approval. An approved drug that patients can't access isn't worth the time, effort, and money that was spent to get it there. Access means sustainable availability, insurance covered, qualified treatment centers, commitment from industry to stay in the fight when the path gets tough. And it's something that I've thought a lot about since writing that first check in 2014. So what has 19 years of rare disease patient advocacy taught me? In May of 2014, during the American Society of Gene and Cell Therapy annual meeting in Salt Lake City, I took a walk with a renowned gene therapy researcher. It was a 45-minute walk from one meeting to another. But the learnings that I took away from that walk have been foundational to my advocacy efforts ever since. I was focused on the science. It's all I thought about, with little thought of what comes next. If I had the science, I'm golden. I'm going to find something for my daughter. I'm going to save her life. Life is going to be good again. But what I heard was, Sharon, you're going to need to do more. You must help prepare a way for the science to move forward and do what it can do. It was that walk that inspired the formation of the nation's first Rare Disease Advisory Council to support understanding and policies at the state level, enabling early diagnosis and ensuring the rare disease community has access to the best clinical care. North Carolina's RDAC was signed into law a little over a year later in August of 2015. Very quickly, I think maybe within a day, I received a call from a rare disease advocate in Minnesota named Erica Barnes, and the rest is history. I consider Minnesota's Rare Disease Advisory Council to be not only a gold standard, but the gold standard. And I dream that North Carolina will follow your lead. Taylor passed away more than 7 years ago. Grief gets easier, but I'm promising you it doesn't go away. What encourages me and keeps me in the fight is that I know there are families like Erica Barnes channeling their grief and their frustration into action, demanding that policymakers, regulators, and industry not only listen, but include the rare disease community in their decision-making. There is so much that only a patient and family know for sure, and what they know more than anyone else is how much they need a treatment. So Shelley Bowen, Barth Syndrome mom and Director of Family Services and Advocacy with the Barth Syndrome Foundation, shared a comment during last November's NORD Summit, and I've shared this comment over and over and over since Shelley shared it with us. In the rare disease space, there is a commonly used phrase that, regardless of intent, marginalizes us from the outset, which is, we need to have a seat at the table. It seems consolatory. To be clear, it is our table. Our people are suffering and dying. We are the experts in our conditions. We know who should be on the guest list. We know the unmet need, and we should drive the discussion. We know the natural history, and we are acutely aware of what the future holds without therapies. So we should set the priorities. We need help. We don't need consolations. True change will not occur until there is a shift in embracing that it is our table. When I began my rare disease journey, I was just a mom, and a mom with no science in her background. I heard piano players, piano performance majors don't need science. I heard that from high school, but what I'm going to tell you is, yes, we do. And we need a lot more when our children are diagnosed with rare and devastating diseases. We'll do what it takes to become experts, and we'll bring other experts into our sphere when we can't get it on our own. So I want to leave you with a call to action. We must sustain urgency at the highest level. Delays equal lives, and we're losing a generation of children. We don't have time. Our children don't have time. We must embrace innovation and say yes more often than we say no. Families know the risk of being on the front end of innovation. They also know the risk of doing nothing. And finally, as we navigate these thorny issues around moving treatments to patients, bring us to the table. The fact is, we are the table, and we need all of you around it. And we'll set a place for you. Thank you.
Thank you so much for that wonderful presentation. At this time, we'll have our question and answer session. There will be students with microphones that will be walking around. So if you have a question, please raise your hand. And then there's somebody with a question over there. So please direct your question to one of the panelists. And I think, hopefully, Dr. Shuck is online. Okay.
I hope so too. My question is actually for Dr. Shuck. My name is Dan Bourne. I'm a co-founder of Helen's Pink Sky Foundation, which, and I could have got through this until you spoke, Sharon. Absolutely amazing. My daughter was diagnosed with Batten disease almost exactly a year ago today, March 10th of 2025, and we realized very quickly that we couldn't just say okay to what the doctors told us was coming, and that we got the same word as you did many years later, which is enjoy the rest of the time you have and make memories. We started a foundation and have been, I would say, very successful. We sponsored research at a number of places. I'm sitting here with some of the researchers from the University of Minnesota who are onto something incredibly promising. And we've actually submitted an IND for an N of 1 trial with the FDA. Through this whole process, though, I felt like I've had a little bit of control. I don't have control over the disease, but I've had control over some of the outcomes, fundraising, who we choose to partner with, how I choose to advocate for my daughter. And, you know, as we get to the FDA, it feels a little bit like it's a black box. I don't necessarily have control anymore. My question for you is, what is the place of the parents when it gets to this phase? I have full trust in the researchers and Dr. Orchard that are helping us through this process, but with the FDA, how can I advocate for my daughter? How can I share how meaningful this is? The natural history of this disease is very well known. My daughter, she'll be 5 here in in a month. She's lost her ability to talk completely. She will go blind. She'll lose mobility. She'll lose her ability to eat. She will suffer from adolescent dementia, become bedridden, and and that will be the end. So how do I help advocate for something that we don't have time with, something promising and something that we need today? That's my question. Thank you.
Dr. Shuck.
Yeah, hopefully you can hear me okay. I got a message that my connection wasn't great, but
No, we can hear you.
good. First of all, thank you for sharing your story, and I can't imagine what you're going through, and we're all human, and we all feel for you, and doing everything you can. I can certainly appreciate that as a parent, and wanting to be able to advocate for your child. In a specific case of like a single patient IND, to answer your question, a lot of that is frankly, like the scientific paperwork and those things. And I was trying to, as you were asking your question, come up with a specific way in your scenario that the parents are involved. And if I'm being honest, I can't think of anything specific that fits that exact scenario. But I will say that there are, a number of ways that patients and parents and caregivers and patient advocates interact with FDA that is highly, highly impactful. From, when a drug is farther along the line and at the approval stage, participating in advisory committees and things like that. Those stories of patients and how they're impacted by the disease and how they're impacted by the drugs are extremely impactful, and that's true for everyone involved. In other ways, there's patient listening sessions and things like that I mentioned, and I don't want that to come across as like the generic seat at the table that was mentioned, because it's really not. We learn immensely from these things. We learn. There's all the speakers I think talked about endpoints. We learn what's important to patients, and we take that back, and we really do apply that to how we can regulate these drugs and everything. So I in terms of single patient INDs, I'm not sure what the role for the patient is there, if I'm being completely transparent, other than being that advocate. But there are many ways in which patients are involved in the process and caregivers are involved in the process, and those do have a huge impact, and hopefully you're able to continue to advocate for your daughter in that way.
Thank you, Dr. Shuck. I feared that was the answer. I'm going to find a way, some way to positively influence. But I appreciate everything you shared and for your time today.
All right, other questions? We can take some from online too, if no, so no online questions. Okay. Other questions from the audience? I guess I also have a question for Dr. Shuck. Recently, Moderna filed for an FDA application for an mRNA vaccine for treating the flu, and that was initially denied without review, and then I guess something changed that happened. I wonder if you could comment on the process that went on.
So I can't comment on specific review decisions, particularly vaccines are not even in the center that I'm involved in, and I'm not familiar with the data that led to that decision, or the change in decision. From what I've read in the news, which is I'm sure where you're getting this from as well, is there was a slight change in the patient population for which the indication was being sought that led to it ultimately being accepted. But if I can speak a little bit in general about the decision to file a drug, which file means for the FDA to give it a full review, as well as the decision to approve drugs, these are extremely complex matters. And that benefit risk assessment hinges on countless different factors that when you see these stories in the news about, FDA said this, then they didn't approve the drug, and that indicates some, overarching change. I don't think that's always necessarily true. there can be agreements in principle that a certain endpoint might support approval, for instance. But we're making, those statements and we're having those conversations, as I outlined, with companies, throughout the drug development process in the absence of data. And when those data come in, it can change the interpretation of things. For instance, there might have been some agreement that an endpoint might be important for patients, but when the data come in, rather than normalizing a biomarker, it's barely reduced, and that changes that conversation in terms of the likelihood that that's going to actually benefit a patient when the magnitude of effect ends up being a lot smaller than what we're anticipating it to be. Or another example is, use of external controls is very common, and we might, in principle, as I shared an example where that was done successfully and a drug was approved. But sometimes when the trial data comes in, that external control doesn't match the patients that got the drug very well, and there's confounders in there that can make it look like, at first glance, a drug had a very large effect. When you drill down, it can be systematic differences in those patient populations that made it appear that there was one, and when you look closer at the data, there's not much there, and those things complicate these a lot, unfortunately. And I wish that every drug that went into patients was effective and worked, and it was clear to see. As has been discussed throughout the different presentations, that's not always the case, and we have to rely on the scientific process to weed out the ones that are unsafe and are ineffective, in order to show which ones are safe and effective, and those are the ones that the patients want to be taking and that can benefit people, and that's kind of the process. And hopefully that answers your question. Again, the specific cases are hard to comment on without knowledge of what the specific data were and what the deliberations were within those teams. But people are weighing a number of different factors, and there's a lot going on in these different applications, and subtle differences can make a big difference when it comes to interpretation.
Thank you. Other questions? We have a question over here.
That's one question.
Dr. Baer, where's the microphone?
Online.
Can we get a microphone to Dr. Baer right there? Yes.
Thank you for the opportunity to ask this question. One of the things I've been engaged with a number of patient advocacy groups and other research teams is the utility of natural history registries as baselines, or evidence that's used to establish efficacy and impact of drug trials or medications or interventions. And there's a lot of variation, obviously, in the way those data were collected. They're not set up where you've had a big review or a systematic analyses of the quality of the data, but they are meaningful and broad and sometimes very long. How can we use effectively these longitudinal data sets, for example, from the Rare Disease Clinical Research Network, or family-supported interventions such as the I Am Rare registries that NORD has been supporting, so that we can help family groups and researchers use existing data sets that could be effective in helping to establish the utility of new drugs? So your comments on that kind of data as evidence or as provision of natural history. Thank you so much.
Somebody want to address that? Would you like?
It's a general question to
Okay.
any of you who are trying to bring new drugs to market or thinking about new drugs, because there's been a lot of emphasis in the rare disease community to try to gather this longitudinal information for exactly this purpose. And if it's being gathered with no sense that it will accomplish that, then we need to think better about how to do it.
You have a microphone.
Well, I can say from the MPS perspective, everything we do, I said that we're drug to child, but we know that that requires data. We have a model family support program called Pathways. I'm not sure if there's another group across the country that actually has a program like Pathways. This program was put into place It was to support our families at diagnosis, but it was also all about data gathering to move drug to child. And what happens is we have social workers and genetic counselors who work full-time for us, but they're stationed in different parts of the country. We have one in Denver, one in Boston, one in the North Carolina area. And when a family is diagnosed, when we receive word, these social workers, genetic counselors go to the family. They'll get on a plane. One last week drove 8 hours on a Saturday morning to get to a family who had just recently been diagnosed. It's not just that early days of anxiety and helping families match up the services and the things that they need to move forward with this new impact in their lives. It's also about what we learn to inform, gathering that evidence to inform drug development. We also just inherited a partnership through Sanofi. We have just taken on the MPS registry, and we'll be building that out. We understand the impact of incredible, well-developed data. And we actually will have someone who is within the society who is driving that data. That's what they do. This person's job will be to make sure that this data qualifies, that it's good data to support good science and good treatment development.
I would like to hear Dr. Schuck comment on this as well, and I think the challenge is in what is the data, what's in it, I think, and we haven't moved forward in a clinical trial with historical data from a registry, so I can't speak specifically about it, but I think first questions are going to be, what is the variability in your population? What are really the time to event data that are going to inform how these patients progress over time? How different do they look? To know what to expect in your trial. That's going to help you set endpoints. And I think it's those endpoints that are going to be critical, and it's tough to get those without a prospective study, is what I've learned. I think it's likely going to depend a lot on that disease, but if you're doing prospective outcomes assessments that are not done routinely in clinic, that's likely not going to be in your registry data. So if you're selecting those as endpoints for your trial for effectiveness, then it's tough to use the registry data to have that present, right? But I think those are the challenges. But I would really love to hear Dr. Schuck's comments on using that type of data for effectiveness evidence.
Yeah, I'll do my best. I'm a clinical pharmacologist, and natural history studies are certainly not my core area of expertise. So what I say will probably be a little bit generic, but for one, this is actually an area that I think that the community has made massive strides in. When I was first starting at the FDA like 13 years ago, natural history studies were if you were lucky if you had anything at all, and they weren't really conducted with this in mind. So the fact that everybody's proactively thinking about this right now is, I think, leading to them being more consistent, more thorough, collecting more meaningful data that we can actually use on the back end, and I think that has made a big difference. And I highlighted one example of an external control being used in a clinical study to support approval, and there are other examples as well. Making sure that you have, broad representation that's going to match the clinical trial population, I think is important. The tendency here is to say, collect as much as you possibly can. And I, what I want to say as a word of caution is I've been in these patient listening sessions that I was mentioning before, and heard about the burdens of being involved in clinical trial research and how impactful that is to families. And I do think it's important to balance those things. So I think that anything that's included in a natural history study, especially like those extra assessments like you're mentioning, really need to be done thoughtfully, and not treating patients as pincushions and expecting them to, fly across the country routinely to do these assessments. So I think one thing we can do as a, research and scientific community is figure out ways to do these things better for patients, use of remote technologies and things like that could help us collect those data in a way that's convenient for patients, but also, done in a rigorous fashion that can be used for regulatory purposes. So, those are the kind of things that I think we'll continue to get better at. But I do think just that, rigorous systematic assessment of data has drastically improved, and continuing to do that while thinking about how a piece of information might be needed and what the burden is to the patients and caregivers to collect that can help design, effective natural history studies that can ultimately provide very valuable information.
Thank you for that response.
There's one
And
question online.
we have another question.
There's one question
Oh.
online. Can you hear me?
One question online, okay?
Yeah.
Can you repeat it?
Yeah. So it's a question from Robert Kessler. He's asking to Ms. Sharon King and other parents who have walked alongside children with degenerative conditions. When you reflect back on the innumerable interactions you had with healthcare providers during your family's medical journey, what made interactions particularly encouraging or empowering? So Dr. Kessler, he is an early career provider, and he would love to tailor his interactions to be as supportive as possible. And there's a note that he specifically is in the context of discussing potential treatment options with no guarantee of success.
Oh, Dr. King. Let's hear.
Was that an echo?
Could you maybe repeat the question? And
I think
that's more
it's
succinct.
basically, Dr. Kessler is an early career provider, and he wants to know how to better support families, especially during discussions around treatment options with no guarantee of success.
That's a really good question, because in my personal experience, I know that I was bound to find something. If there was something out there for my daughter, I was going to find something. I had the support. So from my experience, our first neurologist, the neurologist who actually came to the point where she thought there's something here, and we've got to go deeper. We need genetic testing for this child. So I give her credit. She was the diagnosing physician. Yet she called me on day 2 and said, I don't feel qualified. I don't think I can continue in your daughter's care. You're going to have to find an academic medical center. It was good advice, but for a parent who's just received this devastating information to think, our doctor is walking away from us, I think for me, I wish we'd given it some time to learn together, to talk together, to spend some time about what next steps should look like, instead of just jumping to, this is where we're going to conclude, this is where we need to move. And then from the pediatrician standpoint, who was with us throughout the rest of my daughter's life, we made the decision that when she passed 18, she would definitely stay in the practice of our pediatrician. But I will say that she was, my daughter participated in a novel clinical trial, and I was ready to move. This represented hope for us. I'd done my due diligence. I believed it was the best step for us. And my daughter's doctor called me one afternoon in tears, crying. I'm really frightened for you to do this. I think it's really a partnership between families and their providers and how they move forward. And it's really time intensive, but I think we need to allow for those conversations that will happen not once, not twice, but multiple times. It's like informed consent. You can't consent somebody one time and expect it to be the be all end all. You're going to need to hear over and over and over and over about why this trial, what's it going to look like, expected outcomes, everything that exists within consenting a patient. It happens over and over. For a mother who was living with high anxiety, you set up walls, and sometimes you hear what you need to hear, and you don't hear what you can't deal with at the moment. I think the same thing is true of how we work between families and providers. It's got to be a strong partnership relationship. I will leave this comment by saying that my brother is a surgeon, and he gave me one of the best pieces of advice early on. There would be questions, I would hear, we're just going to do this. This is what you need. The example I can give you was the placement of a feeding tube. There were two options. One required a second surgery, and where my child would be anesthetized. I was really nervous about a child with cognitive impacts, neurocognitive, getting anesthesia multiple times. And it was just made the decision, that's what we're going to do. We're going to do that feeding tube. They literally came to take my daughter to surgery. And I went, you haven't answered my question. And why would I choose this over this? Give me why. I called my brother and he said, I told you, flip the question. The question is, if this child right here was your child, you need to tell me what you would do. What choice would you make? They sent an anesthesiologist up. And I looked at him and I said, this child with Batten disease was your child, what would you do? And he went, I'd do the feeding tube that required anesthesia once. And I went, reschedule. We've just made our decision. I think that's how we work together. Everything about rare disease has felt, it's all about a relationship. From day one, it's how we form the relationships that exist around this patient and family, and how we give them room to breathe, to understand, how we communicate, and how at the end of the day we make joint decisions about what's best for people, for individuals who are living with these incredibly complex diseases. And remembering that half or more of them are children, and that for a parent with a child diagnosed with a complex illness like this, it makes the foundation for discussion. It changes the rules. And so I think as a provider, be really sensitive when you're working with a family that's dealing with one of the most devastating pieces of knowledge they will ever in their whole life face. Because they need your support just as much as that child, as that loved one in their family who's just been diagnosed. That was a long answer, but I've learned that every bit of this is about relationships and how well you connect them.
Yeah. Thank you for that response. Okay, all right. I think we'll close this out. I'm going to thank all of the panelists today for your presentation, and again, it was a wonderful, Now, I'd like to invite Lisa Coles up for the awards for the posters. Here she comes. Okay.
All right, I think I'm wrapping this up. So it is my pleasure today to announce the poster awards that really recognizes the individuals and their contributions to the understanding and or treatment of rare disorders. These awardees are young and junior investigators, students, trainees that presented posters here today. And I'll just say it was extremely competitive, and there were many people who have made comments on the excellent quality of the poster presentations today. So really, I want to acknowledge all of you that were able to present here today. But we have, let me see, I think, if I move this. Okay, so we have 3 awardees, and I'm going to call their names, and when they hear their name, if they could just come up and accept their award. The first is Christina Jackson, and the title of her research is Gene Therapy for Cockayne Syndrome B and Establishing a Stress-Induced Mouse Model. Congratulations. The second award is for Jacob Lundquist, and his research was a novel deep learning approach for automated MRI severity scoring in cerebral adrenoleukodystrophy. And last but not least, the third award is for Julia Jordan, and her research was entitled Getting to the Heart of the Matter, a case study of neurodevelopmental effects in tremelimumab in LZTR1-related Noonan syndrome. And if I could just ask for them to stay at the end so we could take a quick picture. Thank you very much.
Good afternoon. My name is Jim Cloyd, and I have the high honor of being the director for the Center for Orphan Drug Research, which co-hosts this program along with the Stem Cell Center. And we must bring this program now to a close, but I think you can all agree that it's been a stimulating day with some excellent science, some heartfelt and eloquent presentations about life, dealing with rare disorders, and the very promising steps that are being taken to get more treatments faster to those who need them. But we can't close until we say thank you. And so, let's see here. First of all, I want to thank the keynote speakers who just did a perfect job of doing what we asked them to do. I don't think it's necessary for them to stand because they either can be seen here on stage or on screen. But thank you all. Round of applause, please, for our speakers. You will also see that I want to thank Erica Barnes, who moderated our really wonderful breakfast session today. Erica, where are you? There she is. Raised hand. And you now know who Walt Lowe and Paul Archard are, and thank you guys for keeping us on focus. This program would not be possible without the support of our sponsors. They are listed here, and I think it's important to note that in addition to these sponsors, we had others that were of a clinical organization. So it's not only biotech, although they are an important component, but we've had other supporters as well, and we appreciate that. And then last, but certainly not least, I want to say a thank you to our planning committee and volunteers. Putting this program together starts, and this is a warning to the planning committee, it'll start in about 2 to 3 months. Mark your calendar. And the planning committee listed here, that's the first 2 columns on the left. We first do a brainstorming session of what's a topic that's of interest to the rare disease community. And I want to emphasize that term, community. We are dedicated to providing support for families and patients and other caregivers. And that becomes a focus to what we want to do for the next program. So we brainstorm, a couple of Zoom meetings, we throw out ideas. Sooner or later, something sticks. And then we start to plan that actual program. We identify topics. We identify speakers. We talk about a format. And then we go on to do the work of putting the program together. And before I go any further, I want to recognize some of the volunteers. They're the individuals on the far right side. And they were the people that helped you get to your posters, put them up, you could get the exhibits set up, run the microphones, and all the other work that needed to be done. Now, having said that, I have one further comment. You will see in bold the name Brianna Carlson. And Brianna, I'm going to embarrass you. Please come to the stage. Marcia? Brianna took this job on last year as we were planning for the 2025 program, and she actually stepped in as another coordinator had left. So she was learning on the job while doing the job, and it was not an easy task. But hopefully, and I think it's fair to say, there was some learning that took place. But she has managed virtually everything from getting the planning committee together to organizing the program, getting this venue, feeding you, parking vouchers, working with the sponsors, and so forth. So it's been, this would not have happened without Brianna's enormous contribution to this program and her very, very capable management of it. So with that, Brianna, this is a heartfelt thank from the Stem Cell Institute and the Center for Orphan Drug Research. And we have a little offer, if I can find it. Yes. A gift card. Thank you. Thank you. So, to all, thank you for joining us today. I hope you walk away feeling better informed and to some extent, a sense of hope that we have made progress and we're going to make even more progress in the future. So with that, we're adjourned. Thank you.